The following is an update on research and study of BBS. The session was led by Professor Phil Beales, Professor of Medical and Molecular Genetics, Wellcome Trust Senior Research Fellow in Clinical Science, Honorary Consultant in Clinical Genetics, and President of BBS UK.
So thank you very much. I'd like to add my thanks to the committee, the new committee, and the seamlessness with which they've moved in. Steve mentioned that Phil and Chris we miss desperately this year, is going to be a hard act to follow. I think Steve has already shown us that he's easily slipped into those shoes, and thank you very much. Right, let's move on. So in time-honored fashion, I'm going to give you an update on the research and work we've been doing around BBS, around Bardet-Biedl syndrome, over the last year or so. And then we'll have some questions and answers after that. Also, just to give you a rundown, I know you've got the conference program in front of you, but some of you won't be able to see that. So very briefly, yep, we're on time at the moment. So about 10:25, we're going to have a talk on cilia and the eye. Then at 10:50, we will have a personal perspective. At just after 11, we'll break for refreshments. 11:20, we'll have a talk on special educational needs. 11:40, management of pain and fatigue and chronic conditions. Just after 12:05, digital accessibility. 12:25, puberty, sexual health and relationships. And again at 12:50, another personal perspective. And then we'll break for lunch around 1:15. And then just to remind you, in the afternoon we have three sessions of around 20 minutes each, workshops, various types of workshops, and we'll mention them again towards the lunch break. Right, okay, so let's think about what we've been doing over the last year or so. So the first thing to say is a warm welcome to Helen May Samira, who's been a long-term supporter of this organization and these conferences. Used to work in my lab many years ago, and reminded me this morning that she is unique in being the only person we've trained PhD-wise to go out and lead her own lab. So she's patting herself on the back there. But well done. Well done, Helen. It was Beth, I know. But I'm joking, Helen. But it's lovely to see you, because last year, this was the state in which you were in. You were in a heavily pregnant state, and pretty unhappy about that, I think, at the time. But we can now say that we have Luke here today. I think he's swimming at the moment, or sleeping. But anyway, I'm sure Luke will make an appearance later on today. Okay, so the clinics. Most of you will attend the clinics. Many of you are obviously new, and you might have only been to the clinic once or so. But just to remind you of the structure of the clinics, this is an NHS, a unique situation, an NHS-funded standalone clinic service based in England, but it reaches out to the rest of the United Kingdom and also into Ireland and the EU as well. We have a few patients from the EU. So, sorry, we're all in the EU, aren't you? I'm Brexit mad already. So the structure is such that we deliver these clinics in 4 different hospitals, 2 in Birmingham and 2 in London. So the ones in Birmingham are Birmingham Children's Hospital, and Tim Barrett is here at the back. Brilliant, lovely to see you. So you'll have an opportunity to speak to them later on. University Hospital Birmingham, where we see the adults, and in London we see the kids at Great Ormond Street Hospital, where myself and I'm not sure whether any of our other clinicians here apart from Kath. So Kath Sparks, our dedicated nurse, is here. Oh, sorry, and Sarah. Anyone else? No. So Sarah, put your hand up. And Kath, where's she gone? There she is. So just so you know who they are. And Kath works also with me at Guy's and St Thomas's, where we see the adults in London as well, or rather south of Birmingham. But it's a unique service. It's supported in a unique way by a dedicated genetic service to do genetic testing. And that's a model that is essentially being copied now by many other services. So it was the first to do that. And more significantly, I would say, is the way the whole service has been glued together is by this organization, by BBS UK, as we call it now. And we had to, back in 2010, when the clinic started, we had to put together, or rather incorporate or limit a company so that it could work as an equal partner with these other 4 trusts and the NHS. And it delivers amazing pastoral care and organization of those clinics, which is a model that's being held up by the NHS for many other services. So well done. We're almost around 500 patients now attending the clinics since 2010. We continue to climb, so it's a challenge for us as well as a joy at the same time. So let's talk about probably the main development that has happened in the last year. Last year I talked to you about the possibility of us implementing a patient or a clinical support tool, if you like, an electronic system. And I can gladly say that we've now been able to deliver this. And company Certus, who came and presented last year, I think it was last year, and Ian Bamsey and his team have built us a system which we've now managed to roll out in a couple of centres. We're rolling out across all 4 centres. And I'm just going to briefly run through it just to give you an idea of what it does and where we're going with it. So the idea is that we didn't know what was going on in other clinics. We didn't exactly know all the information around each of you who attend those clinics. And we needed somewhere to store all that information and easily access it. Because you know how hospitals tend to lose notes, all of that kind of thing. So what they designed was an online, in the cloud, a safe, secure cloud, as safe as the NHS's cloud, probably safer in my view. But the beauty of it being in the cloud is it means we can access the information, and eventually you will be able to access that information as well about yourself from that cloud. And I'll talk about that in just a moment. So here's the first page. I'm showing a screenshot here of if you log in to the system and you go into the BBS clinics and you click on a particular date. This one is a Gosh clinic. You'll be able to see all the patients that have been registered to come to that particular clinic. And we can click on one particular patient. In this case, it's Josiah Wedgwood. And we see immediately 4 different tabs come up. The first one is a summary about that particular patient, their address, date of birth, gender, NHS number, email address, telephone, who is their primary contact. In this case, it's the biological mother, Janet Wedgwood, and also their referring practitioner. We then move forward and we have this very helpful timeline, which is on the next tab. And the timeline tells us at a shot, a snapshot of exactly what kind of visits or interventions that particular patient has had with the clinic. And so it's very difficult to see from the distance, but just to describe, under 2015, for example, this particular patient, Mr. Wedgwood, has had an endocrine assessment. And what we can do is we can click on the endocrine assessment and it will bring up a form, and it will either give us information about the last assessment, or it will allow the endocrinologist, in this case, the hormone doctor, to add information about that particular patient on this particular visit, and then that gets stored with the various results as well. And we do that for all of the 7 different specialists who see the patient on the day. We can then move to the next tab, which is the actions, and we can see in this case that the clinic administrator has set up an appointment for this particular person. Next to that, there's a drop down, and we can do a number of things from that. We can add various other clinical assessments. We can add blood result tests, or we can print the clinical summary, or we can even send out an appointment letter to you, all automatically. And so this is just an example of the clinic letter that we might send out that's generated from this system. And this is an example of, and it's very much the same as the letter you already received, the multidisciplinary team letter, which is about 2 or 3 pages worth of information summarizing all of the actions and discussions that you had with the ophthalmologist, the nephrologist, kidney doctor, the hormone doctor, the dietitian, the geneticist, the psychologist, and the speech and language therapist, and anyone else you might see on that particular day. So this is a real benefit to us because it speeds up the letters being developed, but keeps all the information safely in one place. And the last tab is a log, which essentially tells us all the things that are particularly happening, all the events that are booked or have happened or are currently happening for that particular patient. So in this case, Kath has made another appointment for this particular patient, Mr. Wedgwood again, but in that we can click on her name, or next to her name, a little icon, and it will bring up a forum message as a messaging tool, and we can send a message about that particular patient to Kath and share that with other people as well at will. So it's a sort of a discussion board, a safe discussion board about that particular person. And finally, this system is now rolled out on tablets. We like to remain in the 21st century. So we're using tablets at Great Ormond Street. I think next week or the week after we're going to be doing the same at Guy's Hospital. And I'm not quite sure about what the situation is in Birmingham. But every clinician, every specialist gets to have an iPad or some other similar tablet, and the information is all there at their fingertips on the day of the clinic, and they input the information there on the day of the clinic. So it's all almost real time, if you like. Okay, let's move on from the clinics to the genetic testing. And this is something, as I mentioned earlier, that is been pretty unique to our service, because when we first set out, well, certainly in this conference, many years ago, very few genes had even been discovered. We found the first gene in the year 2000, and now there are around 19 genes, and you only have to have 2 mutations in any one of those genes to have or develop Bardet-Biedl syndrome. So therefore, we've been able now to develop genetic testing, the first genetic testing of its kind, certainly in the NHS, and I think probably the first in the world, actually. And this has really been enabled by Beth Hoskins, who you know well, who's developed all this test as we go along. I found this selfie picture of hers on the Google Images, I think. So she obviously likes taking pictures of herself. But she's been instrumental in delivering these particular tests. She's a clinician scientist down at Great Ormond Street Hospital in the genetic testing lab there. But I'm not sure what else she does, because she seems to have quite a lot of time to to take up a new career, which is a modeling career. And those of you who are profound readers of The Good Housekeeping, particularly in March, will come across this sorry, I almost said full frontal, didn't I? Those of you who can't see, like Steve, it's not a full frontal, okay, of Beth. It's a full page spread, is what I was trying to say, around Beth's activities. But on a serious note, she's really embarrassed now. I'm sorry about this. I'm not mean, because this is really on a serious note what Beth does. So she works with or is part of Good Gym, and that organization goes around with lots of other people, I think run around with lots of other people, doing good causes, um, sifting through food banks. You want to say anything more about that? Or, yeah? And generally doing great causes, as well as her love for exercise and running and doing marathons and various fundraising events. I don't know how many thousands of pounds she's raised for this organization over the years. But I just want to say thank you. Congratulations. So back to serious matters. Beth put together the latest figures for those who attend the clinic. 376 of you have been tested who come to the clinics. And so far, of the genes that we have tested, 82% of you now have the mutation confirmed in one of those, well, actually one of the 12 genes that we actually test for. And that means there's a few left over to be discovered. And that's sort of ongoing, if you like. But it's important because it allows us to do carrier testing. And 164 relatives have now had a carrier test. And this graph is just showing how common the genetic mutations are in the various genes for the carriers. And as we always see, BBS1, many of you know this, but for the newcomers, BBS1 is the gene in which there are found the most mutations in the UK at least, and North America as well. In the Middle East and other countries like that, it'll be different genes that are much more commonly mutated. So BBS1 is closely followed by BBS10, and then that's followed by BBS12 and then BBS2. And that's very valuable for us knowing this information, as well as for you providing carrier tests. And equally, we've been able to offer you, and 18 of you have taken up prenatal tests. So that's tests in the womb of an unborn child to determine whether that child is going to be affected or not affected going forward. And we always say this, but this has been one of those real success stories, is that when we first started, there was a survey we did with this society back in 1999, which we published with Drina Parker, and then the average age of diagnosis was around 14 years of age. It came down a few years ago to about 9. But what we've been able to do is, through testing and through the service and your engagement, this age at diagnosis is getting lower and lower. And that's because together we've been able to raise awareness of the condition. Many of you, we've had lots of discussions at this conference in the past about how GPs, doctors, as well as the general public, just have never heard of Bardet-Biedl syndrome. It's still an uphill battle, but it's a hell of a lot better than it was several years ago. Right, let's move on to some of the research particularly, and this is just been doing in my lab in London, and we're, as ever, focusing a lot of our attentions, not all of it, a lot of our attentions on treatments, and the three areas where we like to, or are working on, are developing medicines, if we're looking for medicines that might help to alleviate some of the problems that you have living with Bardet-Biedl syndrome, using, for want of a better term, gene engineering type, or gene medicines, should I say, and also gene therapy. And we'll talk about just one or two of those in just a moment. Gene therapy is about replacing the broken gene, or when I say replacing, it's adding the broken gene back. And this is something that Victor, who's here today, where's Victor? Hands up. So he'll be in the workshop again this afternoon. We can talk a bit more about this. So Victor's been leading the charge on developing gene therapy using BBS1, because that, as I say, is the most common gene that is mutated. So we start with the most common one. And he's taking a human BBS gene and sticking it into a mouse. And what he's doing is injecting it into the back of the eye of a mouse that has Bardet-Biedl syndrome. So this mouse hasn't got a BBS1 gene that's functioning properly, just like some of you. And by injecting the human form of that, he's had some pretty astounding early results. So this is just showing you a very brief picture of the summary of the back of the eye. The back of the eye contains these very important cells called photoreceptors. Many of you will have heard of rods or cones. They receive the light, obviously, and turn them into an electrical signal which goes to the nerve and to the back of the brain, to the occipital lobe. And clearly in BBS, we have a problem with the rods and the cones, where they're not working properly. They start off working very nicely at birth, and then they deteriorate for whatever reason. And what has been shown in other conditions, that it is possible to replace some of the genes and restore or retain a certain amount of function in those photoreceptor cells. And that's what Victor's been trying to do. So what he does, he will inject one eye of the mouse and leave the other eye untreated, so you have an internal control for that particular mouse, and then compare over a 6-month period what happens on the ERG. So you remember, all of you will have had, who go to the clinic to see the ophthalmologist, will have had the electrical test where they put a little electrode on your forehead, maybe on your eyelid, and measure the electrical responses going through the eye, going through the retina. You can do the same thing in mice. So again, this is what Victor's been doing, and in short, what he's been able to show is that the eye that's been treated with the gene therapy, with the BBS1, will retain its function compared to the untreated eye, which continues to degenerate. We've completed a first round using 13 mice over a 6-month period, and we're on to a second round now, because you have to prove that all of these things are very safe and not toxic and all these kind of things before we can even consider moving into man. So the second round is at its early stages. It's just beyond the 1-month period, and this is looking even better. There looks to be a very significant amount of rescue or improvement in the treated eye compared to the untreated eye in these 6 mice. So we've got a lot of work to do yet, so we're not making any promises because there's a lot of regulatory framework to get through, and as usual, it's about funding. We've got to get some funding to be able to go to trials, to human trials. Okay, the next piece of work is based on the Nobel Prize that was presented to these guys in 2012. The guy on the left here with the crazy haircut is John Gurdon, and the guy on the right here is Dr. Yamanaka, who's a surgeon from Japan. And both of these guys have essentially created a revolution in the area of stem cells. So John Gurdon first described stem cells back in the 1960s, but Yamanaka has created a real revolution just in the last few years in biology. And what he essentially determined was that you can take a piece of skin or any cell in your body, turn it back by adding a chemical mix, should we say, into stem cells. So for those of you who don't know what stem cells are, these are cells that have gone right back to the status of a very early embryo, if you like. So they can become anything. They're what we call pluripotent. So those stem cells can become nerve cells, they can become liver cells, they can become heart cells, what have you. So what he showed was that cells that have already got specialist function, and we usually use the skin, or we can take cells from the nose or from the hair or from the blood, for example, treat them in a particular way, they become stem cells, and then they can become any other kind of cell, almost any other kind of cell, if you want to look at those. And what is the value of this? Well, it's because there are many aspects that we can't understand or can't test in the lab, because we can't get hold of retinal tissue, for example, or bits of brain or nerves or these kind of things. But this way, we can do that. And so, in our lab, we've been able to start to use this system, and this is work from a PhD student of mine, Rosie Davis, who has really been working and reformulating the various recipes that are out there to take skin and make an eyeball from it. And I'll show you that in just a moment. So many of you, in fact, I think 56 of you, have been very kind enough to offer a small skin biopsy, which we've taken at clinic. And I just wanted to tell you what we've been doing with those particular bits of skin. So we take the punch biopsy from you. We then took those cells to the Sanger Institute in Cambridge. They turn them into stem cells. They then send the stem cells back to us. And then we have developed protocols that are already published out there, and tweak them to turn these stem cells into photoreceptors, into retina. And that's exactly what Rosie's been able to do. I apologize for those who can't see this, but this picture in the middle here is after 28 days. We start to see a lot of neurons, a lot of brain cells, and then we start to see these round football-like structures. And after 70 days, these structures float away from the surface of the dish in which they're growing, in the nice incubator, and got lovely what we call media in which they're feeding off. This is a rudimentary eyeball. It's what we call an optic vesicle. When we look inside it, we can see all the layers of the retina. So that's amazing, isn't it? This has come from a skin biopsy we've taken from a particular patient, and we can do this for anyone, doesn't have to be a patient, and grow an eyeball. I mean, that's just amazing. And why are we doing this? Because we want to better understand what's happening, in your eyes. And we also want to know and have a system where we can add medications in order to see whether we can slow down the degenerative process. Some of you will also be pleased to know that we do something with the urine as well, because we've asked some of you to pee into a yellow pot for us. And that isn't just because we're interested in collecting your pee for looking to see what's in it, but we're actually interested not in the yellow part, the liquid part. We're interested in the cells that are floating around in that urine, because they've come from your kidneys. I mean, urine obviously comes from the kidneys, but they take cells with them that are shed from the tubes in your kidneys, what are called the tubules or the nephrons, and they collect in your bladder. And then we take a fresh sample. We have to have it fresh. We have to process this within 4 hours in the lab, and then we have to grow them in the medium, in the incubator, nice warm 37 degrees, for 4 weeks. And those cells, what we call proliferate, they cover the bottom of the dish, and they divide. And then we can use them in a couple of ways. We can look to see what kind of other genes are being what we call expressed. In other words, which other genes there are that, compared to someone who doesn't have Bardet-Biedl syndrome, might give us an idea as to what's going on as far as the kidney disease is concerned. And then another assay we've developed is called 3D culture. So what we make are these spheres. So if you leave these cells for 2 to 4 weeks, and then we take them off that particular sticky plastic plate and we put them onto a jelly-like material, they will automatically form into a ball, which looks like a football. It is just like a football. You've got the skin on the outside, if you like, of the football, and on the inside there's a gap. It's empty. But poking into those, into the emptiness of the inside of the football are cilia. We've talked about cilia previously, and Helen will tell you more about cilia in just a moment. But the beauty is that Katia, another PhD student of mine, has been able to look at the properties of these particular spheres, these spheroids as we call them. And you can see one here that is, there's a 3D image of this football that's spinning around, and the red lines here are actually cilia. But the beauty is that we've now been able to, sorry, this is a busy slide, so ignore it, but the graph here I'm showing is all of the patients that we've looked at so far. All of them have abnormal spheroids, or develop abnormal spheroids. They're much larger, so they're like a big swollen football. And so this is the first time we've been able to see an assay that is robust assay, a robust test of what's going on in the kidney just by you peeing into a pot. So this is a major advance. You don't have to stick needles into your kidneys, biopsies, all of those kind of things. We can just get you to pee into a pot. So this has been really helpful, and this now gives us a test or an assay that we require to test a number of drugs that I described last year that we've been using zebrafish for. We found a number of drugs. These are already approved drugs out there that seem to have a benefit on kidney problems. Okay, so they can prevent, we think that they can prevent kidney cysts forming, those kind of things. And using this particular test now, the next thing in the next few weeks we're going to do is add those drugs that we took from the zebrafish study to these particular cells to see whether we can prevent them becoming large and swollen. So hopefully next year we'll be able to give you the results of that. And then the last thing I want to talk about very briefly is something I introduced last year, which is the study of Sonya, who's not here today. She came last year. But, this is where she's been studying, again, BBS mice and been looking at their brains. And she's made a pretty astounding discovery, and that is that in the brain of the BBS mice, the number of branches of the brain cells, so you've all seen in those images of on the TV and in magazines of how nerves are like branches of a tree. There are much fewer branches in the BBS mice, and on those branches there are little things poking out called spines, where they make the connections to other nerves, so what are called the synapses. You've probably heard of those. So these are the interconnections that are happening in the brain, and they seem to be reduced in BBS mice. But furthermore, what Sonya has been able to do is show that a very simple intervention significantly improves the connections and the number of branches that happen in the brain of those mice, and that's exercise. So normally our mice, they're lying on their couch watching TV with the remote, eating popcorn or whatever it is, and so what we do is we put a little wheel into their cage and take away their remote control, and amazingly they're so bored they get up and they start doing some exercise. And after just a few weeks, when we have a look at their brains again, we see a complete recovery. In other words, it's gone back to normal, the number of neuron branches, should I say, of those particular brain cell trees. And also the number of synapses have increased as well significantly. Now all of this we know, because we published this with you guys previously, goes on in a part of the brain called the hippocampus, which is important for memory and learning. And so what we want to do, and this is a new study we're starting over the summer, in a handful, it's a pilot study, so in a handful of children, we want to introduce brain scanning and an exercise program. So what we're going to do is we're going to recruit a handful, about 20 children, put them into an MRI scanner, as we've done before, do brain scans on them, look at their hippocampus, then put them onto a 6-month exercise program, which we've worked out with a physiotherapist, and this will be done at local gyms, and the trainers will give these kids a certain exercise program to do. At the end of the 6th month, we'll do a second scan to see whether there has been any improvement. And this has been done not in BBS before, but it's been done in several other studies. We all know that exercise is good for us, but it's also good for our brains. And this particular study from the University of Illinois simply shows 20 students who've been taking a test after eating quietly or after a 20-minute walk. Simple exercise. And you can see already the activity in the brain is significantly increased in the learning parts of the brain after that 20-minute walk compared to just sitting around. So just a small amount of exercise should have an amazing amount of change on the brain. Okay, so I think time's almost up there. I just want to finish up by saying that Elizabeth Forsyth, who couldn't be here today, has just had a baby Otto on last Monday, and so she says hello, and we say congratulations. So I'm happy to take any questions briefly. I think time for 1 or 2 questions. Steve, just wait for the microphone.
The software you were talking about earlier on, Phil, would that be, obviously it has to be safe and secure, but would that be possible to be linked up with GPs kind of IT systems in the future, do you think?
I think it's unlikely that we're heading towards integration with the GP systems, because we had a long debate over making this a standalone entity, and there are still arguments why it shouldn't be a standalone entity, but a lot of the trusts, well, as you know, a lot of the NHS hospitals don't have electronic systems, and some do. Birmingham has one of the best ones in the country, for example, at University Hospital Birmingham, whereas we had a very rudimentary one at Great Ormond Street. So they can't even speak to each other. So that's why we decided to develop a third party on its own, and we had quite a lot of resistance for that. But where I see that the interaction with the GPs can happen better is because in the next version of this, what we're doing is developing a system so that you, Steve, or anyone else can look at their own information. So if you happen to be on holiday in, I don't know, Disney World or what have you, and you have an accident or something, God forbid, and you have to go to the hospital, you can pull up on your iPad or whatever you've got, tablet, or the doctor can do it on their own system with your protected password, and look at all your information, see your results, hopefully in the future see some of the scans, those kind of things, and your latest blood tests. So that way you can do the same thing if you go and see your GP. So I think it will supersede that issue. Go ahead.
That student who's growing the eye,
Mm-hmm.
is there any reason why what's happening could you grow a complete eye? So take the old eye out, put the new eye and connect it in. Is that
I knew you were going to ask that, or someone was going to ask that. Yeah, it's very early days. I think I can't see that happening certainly in my lifetime. But
could you grow it to what we recognise as an eye?
no, it's simply and one day it probably will happen. But there are just too many interdependencies. So dependency on what's going on from the bone. As the bone develops around the socket of the eye, and the eye's developing in there in the fetus, there's lots of messages that are being sent to the eye, and those are absent in the dish. So, I think we're getting to the.
She can, without any
That's right. So she's got as far as she can. And this takes 150 days. This is half a year, really, of her life. She won't let anyone else touch these cells or go near them because they're like babies to her. But you can imagine, it takes 6 months.
So the next thing will be to, as it were, grow it, see how far you go under restraints.
So far under restraints, we're not going to take them beyond 150 days because we know that they will just die after that point. Yeah, good question. Okay, time's up. Being told. Just last thing, last comment is that Kath has put together a questionnaire on the future of BBS research. We want you to tell us where you think we should be doing research, and what we need you to do is fill out this form. They're dotted around all over the place, and there are red boxes dotted around out in reception where you can put this form, and we'll have a look at those afterwards. And then lastly, I want to thank Sonya very much for leading the charge on the new patient information booklet, the medical booklet, which the first one we wrote probably about 20 years ago with Drina, and now this is fully up to date, lovely, glossy, and has a huge amount of information. It's available online as well. So again, you know, there's no excuse when you go and see a doctor or anyone else for that matter. You can just pull that off, give it to them.